Yeah, that's perfect. Yeah. Okay. Thank you very much for coming, and I would like to thank the organizers for the opportunity. So I would like to talk about my paper, Combinatorial Approach for Analyzing Intra-Tumor Heterogeneity. I'm actually currently a postdoc at Stanford University, but this is entirely my work at Brown with Ben Raphael and Ahmad Mahmoudi. So I'd like to begin with showing this figure from a Nature paper in 2012. This is a figure showing an AML patient, showing the cancer evolution in an AML patient. So the cells on the left, the green cells, are normal cells, but then we have some somatic mutations. Somatic mutations are the mutations that are accumulated during the lifetime of an individual. And basically, cancer is driven by these somatic mutations. So as you can see, we have a green cell on the left, accumulated some of these mutations, and then later on, we have waves of accumulating new sets of mutations. So at the time of the initial diagnosis, those researchers took a sample and found out there are several distinct subpopulations that have different sets of mutations. So we have a heterogeneous sample. Later, they looked at a sample after performing chemotherapy and showed one of the subpopulations survived chemotherapy and later gained additional mutations, expanded, and led to relapse. So it's very important to identify these subpopulations. These are fractions of a cell with different sets of somatic mutations. So with this, I'd like to give an outline of my presentation. It has four main parts. The first part is basically an introduction of using deep sequencing to infer these tumor subpopulations. The second part is the main contribution of the paper, using a binary tree partitioning to infer a tumor phylogeny. Later, I will talk about some experimental results, and finally, I will conclude with related and future work. So say we have a heterogeneous tumor sample. When we do sequencing using next-gen sequencing, we lose a lot of information. So we have a mixture of cells, and then the DNA is fragmented into many overlapping pieces. So you don't really know where these fragments came from which of these cells, or where on the genome they belong to. So we lose a lot of information. So we can do resequencing. We can align all these short reads back to reference genome and use the concept of variant allele frequency to estimate the frequency of these subpopulations that have different sets of mutations. So say we are looking at only single nucleotide variants. So I'm showing one of them here. We can align all the reads, consider the reads that cover that single position, and look at the fraction of the reads that agree or disagree with this allele on the reference genome. So say among these 9 reads, 3 of them agree on a variant allele, and the other 6 agree on a reference allele. So this fraction is called variant allele frequency. So obviously, here I'm assuming some assumptions. For example, I'm looking at regions with no copy number. And this is also an estimate because the sequencing is not done uniformly, and we have some other sources of error. So this would be an estimate of the fraction of cells that have that particular mutation. So there are some methods that use, so that was only for one mutation. Now assume we have all the somatic mutations. I have this plot where the x-axis is all possible variant allele frequencies for different mutations. So the y-axis is the number of mutations that have that particular variant allele frequency. So say we have a bunch of mutations at 100% and maybe some 60%, and we have some other lower frequency mutations. So we can do some clustering, basically grouping those variant allele frequencies that are close to each other, so kind of to cope with the complications of the sequencing errors and the way it's done, the estimate, and basically infer these different subpopulations with different frequencies. So there are some non-parametric methods available. I'm showing here some of the very recent ones. For example, PyClone from Saurabh Shah's group in Vancouver, or Cyclone from Leading's group in WashU. So they don't really fix the number of these subpopulations, but they do the clustering using a non-parametric, like a Dirichlet process method. So one thing that these methods do not consider is the fact that these frequencies are actually correlated. If you step back to the clonal theorem, these mutations, the frequencies are actually related, and they come from partitioning of a similar cellular population. So for example, in this tree, and this gives rise to a tree, underlying phylogeny tree, basically. So we have mutations that are on different edges of this tree, and then when we sample, we may basically sample a mixture of these, some of the leaves in this tree. So this is actually not a perfect phylogeny problem when we have sequences or we have samples only each single leaf individually. We have a mixture of these leaves, and we would like to sort of deconvolve and find the underlying tree. So with this, I'd like to move forward to the main contribution of our paper, which builds this tree. So the question is, again, say we have that variant allele frequency plot, the frequency of all these mutations. Is there a tree which is basically related to this variant allele frequency plot? So what is the relation between the clonal evolutionary tree and this plot? So we model this with a binary tree partitioning, as we propose in our paper. So we have some mild assumptions here. First, we assume that when we measure mutations, they are all results of a clonal expansion. And then we assume that each mutation, when they happen, they do not go back. So this is we assume the infinite site assumption. So mutations that happen are present all along after this time. And then at each time, at most one of these clonal expansions begins at a certain time. So this gives rise to a binary tree partitioning. So when we would like to assign these mutations to nodes on this tree such that we satisfy conditions coming from these assumptions. The most important condition that we consider in our formulation is what we call children sum to parent, or CSP condition, which I would go more detailed. So say here, this is the schematic figure. So when we would like to find a tree where mutations are assigned to the nodes of the tree with their frequencies, and each internal node, for example, this node at 60%, has a frequency which is the sum of the frequency of its children. So for example, this 60% is the sum of 35% and 25%. So now, again, the input was actually the variant allele frequency plot. So how can we assign these frequencies to a tree which satisfies the condition? So this is, for example, another simple example. This is actually a very convenient example because everything is perfect. Obviously, we have some errors in the estimation of these frequencies, or maybe we may miss some of these subpopulations, so I will talk about it later in my presentation. So even if this convenient model, the problem, so I will give a formal definition of the problem. We are given a list of these frequencies, say L, A1 through AN. I would like to construct a complete binary tree partition, or what we call BTP, which is a complete rooted binary tree. Every node is assigned with a frequency, and all internal nodes satisfy the children sum to parent condition. So even finding a BTP is shown to be NP complete. You can see the paper for the detail of the proof. So we use a reduction from the numerical matching with target sum problem. We defined an optimization version of this problem. So for that, I would like to define some combinatorial objects and show an approximation for this optimization. So say we define a parent and two children in a tree as a triangle where they satisfy the children's sum to parent condition. So obviously the parent has the highest frequency, and if the frequency of the children's sum to that parent, we call that a triangle. So we call two triangles conflicting if either they share a parent or they share one of the childs. So for example, in this example, we have a list. 0.6, 0.4, 0.2 form a triangle, the red one, and 0.6, 0.35, and 0.25 also form a triangle, the green one. But these two are conflicting. So you cannot form a tree where you have both of these triangles in it. So we prove that the BTP problem is actually equivalent to finding m-1 non-conflicting triangles where m is actually the number of the leaves in the original tree. So say, for example, for this list, you have 3 leaves, and if you can find 2 non-conflicting triangles like the ones that I showed here, you can basically find a complete rooted binary tree. So for an optimization version of this problem, where we would like to maximize the number of these non-conflicting triangles, we obtain an approximation. So we got an almost 2/3 approximation for a problem which we defined as max BTP. So again, the detail of this algorithm is in the paper and also the analysis, but it's basically based on a local search. So we start from the empty set. At each we fix a constant t. At each iteration, we replace S triangles where S is less than t with S+1 triangles that are not conflicting. So because t is constant, this is a polynomial algorithm. So we stopped when we couldn't find a collection of t triangles that could be further get larger. So this is a polynomial algorithm because t is constant, and it gives us an almost 2/3 approximation. But obviously, when we deal with real data, we may not get these perfect binary trees. These variant allele frequencies are determined with errors, and perhaps we have incomplete clonal expansions, like the example I'm showing you here. So say the ideal list was 1, 0.6, 0.4, 0.35, and 0.25, but the one that we observed missed this 0.4 and had other frequencies with an error. So now we would like to add some nodes in the tree, which we call them auxiliary nodes, to complete this binary tree partition. But the original tree may not be binary. So we define this epsilon BTP problem. So for a given list of these frequencies and an error vector for corresponding to each of these frequencies, we would like to find the minimum number of auxiliary nodes that can be added to the tree and make it a BTP, a binary tree partition. So obviously this problem is harder than the original problem, the simple problem. But we can use a recursive algorithm, a very fast backtracking algorithm, to solve it for real data. So the idea is to always maintain a partially constructed tree whose all internal nodes satisfy the children's sum to parent condition. So for example, for this toe example, I would sort all the frequencies. I would start with the roots, and then I would look for pairs of these frequencies that can sum up to a current leaf of this partially constructed tree. So I can just walk through this example. And then if at some point we couldn't find a match, we can add one of those auxiliary nodes. So with this, I'd like to move forward to the next part of my presentation, which is basically experimental results. So we did a lot of simulations, basically. We consider all binary trees of size 3 to 9. For each of them, we created 1,000 different samples. We created noisy variant allele frequency data for each of these trees. And first, we used one of these two clustering algorithms, cyclone and just a standard Dirichlet process, to group these mutation frequencies together. So as you can see, when we just use one of these clustering algorithms, we don't really get a good result. So this plot here, on the x-axis, I have the average, I have all the tree topologies, and the y-axis is the fraction of the trees that have the exact same number of nodes compared to the true simulated tree. And this is obviously an average here. So as you can see, using only these two Dirichlet process method, ignoring the tree topology, we cannot get a good fraction. But when we use our rec-BTP algorithm, the recursive BTP algorithm, on the cluster that we got from these two methods, we improved a lot. We also looked at the exact data set from the AML paper, the Nature paper. So this plot is the variant allele frequency plot for the original tumor variant allele frequency paper I obtained from the supplement of the paper. And when I ran both cyclone and the standard Dirichlet process method, I got four distinct clusters. I ran rec-BTP, and I could obtain the full binary tree partition. And when I compared all these frequencies to the original plot using some distances, these numbers are actually closer compared to the numbers in the figure with the original variant allele frequency plot. So now I'd like to move to the related and future work. So just like half an hour ago, Nilgun presented CTOP from Shahinal group at a different room. And I guess after me, Coed Morris will present their great work, Philosop, which was recently published. So this is like a very active field of research. Just in the last year, there are a number of papers, and there are a number of forthcoming papers dealing with sort of similar models using tree topologies to infer these colonalities. So moving forward, I think one of the biggest open problems is to compare these methods with each other. I think we already started during the conference. I talked to many of the authors and co-authors of those methods. So we should compare these methods. With the promise of the single-cell sequencing and multi-sample sequences, we may get better data sets to validate our claims. So I just got this figure from a recent review paper in Nature Method. We also we are also generalizing our epsilon BTP method. We would like to look at multiple samples, where samples are taken from different parts of the same tumor. So we already have one result, which is coming in WABI this year, using a mixture of multi-samples and trying to deconvolve these multi-samples. And we are looking forward to more results and working on this problem. With this, I would like to conclude and acknowledge my postdoc advisor Ben Raphael, and also Ahmad Mahmoudi, a PhD student at Brown, and also NSERC and the NIH for fundings. Thank you very much, and I will be happy to take questions.
Okay, lots of time for questions. Well, I have a question. Could you start clicking back to around slide 20? Yeah, one more. Okay, well, go too forward. Okay. So I don't understand this figure, okay? Okay, rather than so explain to me again why you join these two nodes to this node and not to the auxiliary node, please.
Oh, that's a very good question. So
Thank you.
in our formulation, in fact, actually, you can do this. You can do this.
Yeah,
So
but you didn't, so why?
because so we basically consider all the yeah. So we would try actually both of them. We would first try this one, see if you can find this 58%, maybe within an error threshold. If we couldn't, we go and connect it to this one, which is now already 42%. So you would check all the possibilities. And in fact, I didn't show the details here, but at the end, we generate all the possibilities, and then we have some measure to pick one of them, say to minimize the error that amortized error that we have for the whole tree and things like that.
So really the criterion is you want to minimize the error of the sums.
Yes, but there is always a trade-off. There is always a trade-off between the number of these auxiliary nodes that you add and the amount of error that you allow. Because if you allow zero error, so you don't change anything, then you have to add a lot of these auxiliary nodes. But theoretically, you can always do that, because if you have like m leaves and you add m minus one of these auxiliary nodes, you can always get a full binary tree. But we didn't want to double the size of the tree. We would allow some of these errors. We would get a smaller tree, but then it depends on what is the threshold for the error.
So I have a comment which will date me. Back in the dark ages, when people used to do physical mapping of restriction examples, one of the chronic problems was to determine the physical map of a partial digest. So an integral graph would be formed, and people would compute the most likely sums that best accounted for the ordering of sites, okay, using essentially a fragment and its two adjacent fragments. So did you look at, and that is exactly what you're doing here, okay, but you just layered a tree on top. So did you look at those algorithms?
To be honest, no.
Okay.
I might have seen one of them, but
You might
yeah.
want to go back and look.
Yeah.
Any more questions? Yeah.
Ming Li from Personalis. My question is, for your sequencing, do you study the relationship between the sequencing depths and your sensitivity? Like how sensitive, what coverage you need to get in order to get certain allele frequencies?
Yeah, so the short answer is that you need a lot of coverage, like very deep coverage. But it's all, again, a trade-off between
Right.
how low you want to go with the frequencies and how deep you want to do the sequencing.
Mm-hmm. I mean, do you have any ideas of how, like...
So for example, these examples that I showed you, they were like, the simulation was like 10,000x. And the real data was like 1,000 to 10,000x. So you really need like targeted deep sequencing.
Very deep sequencing.
Yeah.
Okay. All right. Cool. Thanks.
So suppose in your example, instead of 33 and 26, you had 42 and 12. Then there would be ambiguous solutions to the problem.
Exactly, yeah.
And in fact, with a given epsilon threshold of any size, can't you guarantee the possibility of ambiguous solutions for any substantial size tree?
Yes, you may have multiple solutions. So at the end, you have to choose one of them.
So you have
So
no,
even-
other, you have
even-
no other thing guiding the tree structure other than the adding of the sums then?
Yeah, exactly.
All right,
Good
our
question.
next